Welcome to Excellent Grades Academy. This is Dr. Byson AM. Today we're looking at an exciting topic in physiology, which is blood physiology, in preparation for the assessment that is coming. Let's get into this video. All right, so you can register for all medical courses in second year, third year, fourth year, fifth year, up to seventh year on the number that is displaying on your screen, +260 975 497790. All right, so now, what is blood? So we're looking at blood physiology. Blood physiology, a very high yield topic in physiology. What is blood? What is blood? So blood is a specialized connective tissue. Blood is a specialized connective tissue. Now, remember that the body, despite its complexity, is made up of 4 basic tissues. We have epithelial tissue, connective tissue, nerve tissue, and muscle tissue. And blood is a type of connective tissue that is specialized and has 2 components. So it has 2 components. One component is called plasma. So number 1 is called plasma. Plasma is the liquid part of blood. So liquid part of blood is called plasma. Second part is called packed cells. So packed cells, this is a solid part of blood. The packed cells is made up of all the cells that are found in blood, like the red blood cells, which are also known as erythrocytes. Erythrocytes. So mostly in physiology we'll be referring to them as erythrocytes. And then we have your white blood cells. White blood cells, which are also known as leukocytes. Last but not the least, we have platelets. Platelets are scientifically referred to as thrombocytes. Thrombocytes. So in this discussion of blood, we're going to discuss all these parts of blood, and we're going to discuss the parts that make up all of these components. All right, so before we discuss these components of blood, let's look at the functions of blood. What are the functions of blood? Very quickly. So functions of blood. Number 1 is transportation. Okay. So transportation of gases. So blood transfers oxygen from the lungs to the tissues, and it transfers carbon dioxide as a waste product from the tissues to the lungs so that they can be expelled out of the body. Number 2, excretory function. The excretory function is that blood excretes waste products from cells. So cells will make waste products like urea, and blood will take them to the kidneys for excretion. Number 3, it has a nutritive function. Nutritive function in that it carries nutrients from the GIT to the cells. So the nutrients are absorbed in blood, and then after they are absorbed in blood, they are transported to their cells and they get assimilated in the cells. These nutrients are in turn used to provide ATP, which provides energy for the organism. Number 3 is acid-base balance. Acid-base balance. So blood regulates the pH of the body. Okay, blood regulates the pH of the body. In this discussion, we're going to see how blood achieves that function. Number 5 is that it serves as a protein reserve. So when the proteins in the body go low, the plasma proteins which are found in blood are used as a source of amino acids or as a source of proteins. Number 6 is a defensive action. You see that the white blood cells are involved in the immunity of of the body, that protects the body from pathogens that might want to infect and damage and destroy the body. Number 7 is coagulation. Coagulation. So to prevent further bleeding, blood will form clots on your wounds. Number 8 is regulation of body temperature. So regulation of body temperature. Okay, temperature there. Number 9 is the transportation of other substances like hormones that are dissolved in blood and are carried, electrolytes, enzymes are carried by blood. Transportation of substances. All right, so these are the common functions, most of the functions of blood, but not exclusively to these. Okay, so now when you break down whole blood, so whole blood is separated into plasma. So it's separated into plasma and packed cells. Let me write it here. Packed cells. Packed cells. Okay. So now what happens is that blood is put in a test tube. So if this is a test tube here, and then you add blood. So say this is blood that is added in a test tube. If you get blood out of the body, after some minutes, a few minutes, it is going to form clots, or it will coagulate. So to prevent coagulation, we're going to add an anticoagulant. Okay, so an anticoagulant to this test tube to prevent clotting. To prevent clotting of blood. So what we're going to do is that we're going to subject this blood to high velocity centrifugation. High velocity centrifugation for an hour. Centrifugation. So centrifugation is simply spinning the blood in the test tube at a very high velocity for an hour. After this blood is subjected to high velocity centrifugation, it is going to separate into its components. Okay, so it will separate into its components. Okay, so here, underneath here, that's where the packed cells are going to be found. All the cells of the blood are going to be on the bottom of the test tube, and the liquid part of blood is going to be up here. The liquid part of blood is what we call plasma. So plasma is about 55% of blood. So plasma is about 55% of blood. So this is different in males and females. Okay, so it's actually a range that we're going to look at specifically very soon. And then the packed cells are about 45%. So this is what is going to happen if you subject blood to high velocity centrifugation. Now, what you're going to notice is that most of the packed cells here are actually red blood cells. Okay, so the red blood cells will make the bulk of the packed cells. Red blood cells make up 45% of the packed cells. The amount of red blood cells in the blood is what we call hematocrit. Hematocrit. All right, and then this thin portion here is what we call the Buffy coat. The Buffy coat. The Buffy coat is less than 1%, and it contains white blood cells and platelets. Okay, and platelets. So it contains white blood cells and platelets. So always know that in the composition of blood, the platelets and the white blood cells are less than 1%. The red blood cells are about 45%, and the plasma is about 55%. Okay, so now let's look at these components of blood in details by starting with plasma. So plasma. Plasma is the liquid part of blood. Okay. So this is the liquid part of blood. Now, if we say the liquid part of blood, we don't exclusively say it is made up of water only. No. Of blood. It is made up of two components. Blood made up of two components. So plasma, component number one, is made up of water. The water in plasma is about 93 to 95%. That's the water in plasma. And then number two, it's made up of solutes. The solutes in plasma are about 5 to 7%. Now, these solutes in plasma are two types. We've got inorganic solutes. So inorganic solutes. And we've got B, organic solutes. Organic solutes. So the organic solutes are the major part. The inorganic solutes are just about 1%, and the inorganic solutes are just about 1%, and the organic solutes are about 6%. Inorganic solutes contain what we call electrolytes. So electrolytes like sodium, potassium, chloride, magnesium, bicarbonate. So these are found in plasma as well. Okay. And then the inorganic solutes, the major part of it are plasma proteins. These are very important. Plasma proteins, we're going to talk about them very soon. Other things are nutrients. Nutrients like lipids, glucose, amino acids. Other things that are found in plasma are creatine. So creatine is a fuel that is used to provide energy for the muscles. It's referred to as creatine phosphate. And when this creatine is metabolized, it produces a waste product that is called creatinine. So creatinine is also found in plasma. And this creatinine is removed from the body by the kidneys together with urea. All right, so these are the two components of plasma. Water, 93 to 95%. Solutes, 5 to 7%. Solutes, they are inorganic solutes, and they are organic solutes. Okay, now let's look at plasma proteins. The plasma proteins that are found in the plasma. Okay, plasma proteins. So what you should know is that the plasma proteins are made by the liver. Okay, so these are secreted by the liver. They are made by the liver. So the correct term to use actually is synthesized. Synthesized by the liver. There are three classes of plasma proteins. So they are three classes of plasma proteins. Very high yield data. Very, very high yield data. Always in your tests. Number one, called albumins. Okay. Albumins are the most abundant plasma proteins. Most abundant. Albumin has got a molecular weight of about 60,000 units. Okay. So albumin functions as, so the functions of albumin. Number one, it is a carrier protein. What we mean by carrier protein is that substances will bind to albumin in order to be transported in the blood. Hormones will bind to albumin to be transported in the blood. Metals, ions, electrolytes will bind to albumin to be transported in the blood. So they are carrier proteins. Number two, the buffer pH. So they serve as protein buffers of the blood. So in the blood, there are two types of buffers. You've got protein buffers and the bicarbonate carbonic acid buffer. So the protein buffer consists of albumin. The third and one of the most important functions of albumin is that they maintain osmotic pressure. They maintain osmotic pressure. Now, osmotic pressure is the pressure needed to retain water in a blood vessel. The osmotic pressure that is provided by albumin is what we call colloidal osmotic pressure. So colloidal osmotic pressure. Colloidal osmotic pressure. It is also called oncotic pressure. Oncotic pressure. So this is important to know. Osmotic pressure, colloidal osmotic pressure is the pressure that is required to retain water in a blood vessel. So it is the pressure that balances how water moves between the blood vessel and the interstitium. So if this is the blood vessel here, let me just draw it. This is the blood vessel here. You've got your red blood cells here, and then you've got your water here. Okay. The surrounding space, this surrounding space, this is what we call the interstitium. Now, the movement of water between this blood vessel here, blood vessel, and the interstitial space, this is interstitium, is regulated by albumin, which creates a pressure that is called oncotic pressure. Okay, so the albumin in the blood vessel are going to bind or are going to resist the movement of water into the interstitial space. So water will remain inside of the blood vessel. Okay, so if you have a decrease in albumin, it means that the water will be moving into the interstitial space, and you're going to have less water in the blood. So the volume of the blood is going to be reduced. That is what we call hypovolemia. So you can end up with what we call hypovolemic anemia, meaning that your blood volume has decreased due to hypoalbuminemia, which means that your albumin levels have decreased. Now, osmotic pressure. Let me talk about osmotic pressure. Osmotic pressure is made up of two components. One component that we've talked about is the oncotic pressure that is provided by the albumin. So, and then the other one is what we call crystal osmotic pressure. Crystal osmotic pressure. Okay. Now, the value of osmotic pressure is about 290 to 300 millimoles per liter. Millimoles per liter. The value of oncotic pressure provided by albumin is 1.3 millimoles per liter. Crystal osmotic pressure is 298.5 millimoles per liter. Now, the question is, what creates or what provides crystal oncotic pressure? The bulk of crystal oncotic pressure is provided by sodium. Okay, and its anions. Its anions like chloride, bicarbonate. Okay. The other electrolytes or molecules that contribute to crystal oncotic pressure are potassium, also glucose. All right. Now, what is the function of crystal oncotic pressure? Crystal oncotic pressure maintains cell shape, number one, and size. So the shapes of the cells and the size of the cells are maintained by what we call crystal osmotic pressure, which is about 298.5 millimoles per liter. Oncotic pressure, which just regulates or controls the movement of water between the blood vessel and the interstitium, is only 1.3 millimoles per liter. So this is very important to know about albumin. So that's all about albumin. Okay. Now, let's talk about the second plasma protein that we have, which are called globulins. So globulins. Globulins are of three types. So they are three types of globulins. Okay. One, they're called alpha globulins. Alpha globulins. Alpha globulins serve as carrier proteins. So these serve like albumins. They are carrier proteins. They carry substances in the blood. The second type of albumin is what we call beta, denoted like this, globulins. Beta globulins include one molecule that is used in coagulation that is called prothrombin. So prothrombin is a beta globulin. As you are going to see after we discuss the coagulation cascade, prothrombin is used in the clotting cascade. The third globulin, one of the most important globulins, are called gamma globulins. Okay, so gamma globulins. Now, gamma globulins produce what we call antibodies. So they produce antibodies. Antibodies scientifically are referred to as immunoglobulins. Immunoglobulins. Okay. Now, there are 5 important immunoglobulins. 5 important antibodies. These are, number 1, called, let me shift to another page. 1 is called immunoglobulin A. Immunoglobulin A, which is just IgA. IgA is mostly found in body secretions. So body secretions, for example, in tears, in breast milk, especially the breast milk that is secreted in the first days of breastfeeding, which is called colostrum. Okay. Colostrum has got a high quantity of immunoglobulin A. So this immunoglobulin A will go and provide immunity to the baby. All right, it's also found in gastric secretions. So gastric secretions and GI secretions. All right. Second type of immunoglobulin is IgG. Third type is IgE. IgE mediates, so this immunoglobulin mediates hypersensitivity reactions. Okay, allergies, hypersensitivity reactions like allergic reactions. Okay, these are allergic reactions. Okay, and then we've got IgM, and last but not the least, IgD. All right, so all these antibodies are what we call gamma globulins. They are very, very important. Let's look at the third plasma protein that we have, which is called fibrinogen. Fibrinogen is used in coagulation. Okay, used in coagulation. So this fibrinogen is activated to what we call fibrin. Let me just write it down. Fibrinogen is activated to fibrin. Fibrin is what forms the clot. Fibrin forms the clot. After your clot has formed, so if you see the clot, just know it is coming from fibrinogen. It is coming from fibrinogen. So that is very, very important to know. It's very, very important. So those are the components of plasma. Okay. So now there's something I really want you to know, that red blood cells, the amount of red blood cells in the blood is what we call hematocrit. Hematocrit. Okay. Now, hematocrit in men and in women is different. Okay. So in males, hematocrit is about 40 to 50%. And in females, hematocrit is about 37% to 48%. Now, you notice that hematocrit is what determines blood viscosity. So blood viscosity is determined by hematocrit. Determined by hematocrit, meaning the amount of red blood cells in the blood. Okay. What this means is that, okay, so before I explain this, you might want to ask, what is viscosity? So viscosity is just resistance to flow. Resistance to the flow of a fluid due to interactions of molecules. Okay. Due to molecule interaction. You can say due to friction of molecules in that fluid. Okay. So what determines blood viscosity is hematocrit, which is the amount of red blood cells in the blood. Okay. It is also very important to know that blood is 4 to 5 times more viscous than water. More viscous than water. In layman terms, you can say blood is 4 to 5 times thicker than water because of red blood cells, because of the RBCs. So the higher the hematocrit, the higher the amount of red blood cells, the higher the viscosity. There's always an exam question on this. The lower the hematocrit, the lower the viscosity. All right, that's important. Now, if you have higher hematocrit, if your hematocrit goes very high, okay, you have a condition. So hematocrit grows very high. You have a condition called polycythemia. Okay, if the upper limit of the hematocrit goes very high, you have a condition that is called polycythemia. Okay, so polycythemia is physiological. So where your body is trying to compensate for a certain condition that you've been exposed to. But if it is pathological, it is known as polycythemia vera. There's a difference. So if the hematocrit goes high because the body is trying to physiologically adapt to a certain condition it has been exposed to, such as high altitudes, and because there is hypoxia, that condition is called polycythemia. But if the hematocrit goes high because of a pathology in the body, something has gone wrong, then that condition is called polycythemia vera. Okay. Now, if there is polycythemia vera, what will happen is that there's going to be marked increased blood viscosity. What this means is that there's going to be slow flow of blood in the blood vessels, which is going to lead to formation of clots. Formation of blood clots. Now, if clots form, they're going to block blood vessels. There's going to be blocked blood vessels. If your blood vessels are blocked, then you can develop a stroke. If it's in the brain, you can develop a heart attack, which is simply a coronary heart disease in your heart. Or you can develop what we call a pulmonary embolism in your lungs. So polycythemia is vera is very, very dangerous. Let's talk about the opposite. A drop in hematocrit less than 10% will give you a condition that is called anemia. So a drop in hematocrit, a drop in the red blood cells will give you anemia. It will give you anemia. Okay. So that is it about polycythemia and anemia. Blood pH. Blood pH is 7.35 to 7.45. And this is regulated by the lungs and the kidneys. Okay. All right. So now in this second session, let's talk about how is blood formed. So blood formation is what we call hematopoiesis. Hematopoiesis, this is formation. So this is formation of blood cells of all lineages. Lineages means all bloodlines. Okay, all blood lineages. Formation of red blood cells, which are called erythrocytes. Erythrocytes is what we call erythropoiesis. Erythropoiesis. Formation of white blood cells, which are known as leukocytes, is what is called leukopoiesis. Formation of platelets. Okay, platelets, which are known as thrombocytes, is what is called thrombopoiesis. Okay. So now, formation of all of this collectively is what we call hematopoiesis. Hematopoiesis. Now, let us look at how hematopoiesis is formed. So, hematopoiesis in the prenatal stages. Prenatal simply means before birth. It is also known as antenatal. So, in the antenatal stage, blood formation starts at the first month, the first 4 weeks. So what forms the blood in the first 4 weeks in utero is the yolk sac. Now, if the fetus in utero reaches 3 months, so the third month, blood is formed by the liver. So the liver is fully formed and it's now forming blood. Now, in the 4th month, blood is now formed by the bone marrow. Bone marrow. So this is in utero. Now, once the baby is born, postnatal period, what forms the blood is the bone marrow. The bone marrow predominantly the bone marrow of long bones. So the examples of long bones in the body, you have the tibia, you have the femur, and also flat bones like the ribs. Okay. The sternum. So the sternum is what we call the breastbone, the chest bone. Okay. And the vertebrae as well. So these same vertebrae, the vertebrae of the vertebral column, this is what forms the bones. Once these are formed, the red blood cells have got a half-life, meaning that they are able to exist in the blood for about 4 months, which is 120 days. The neutrophils only last for about 7 hours after they are produced. The neutrophils are white blood cells, and the platelets only last for 10 days after they are produced. Okay. Now, all blood cells are produced from the bone marrow. In the bone marrow, we have cells that are called pluripotent stem cells. So in the bone marrow, you've got stem cells. They're called pluripotent stem cells. Pluripotent pluripotent. So these pluripotent, what this means is that they've got the potential to specialize into any blood cell. That's why they are called pluripotent stem cells. Pluripotent stem cells or pluripotent hematopoietic cells. Other literature refer to them as multipotent hematopoietic stem cells because they can differentiate or specialize into any blood cell. Now, for this pluripotent stem cell to specialize into any blood cell, they require what we call growth and differentiation inducers. Okay, they require growth, okay, and differentiation inducers. So these growth and differentiation inducers can be hormones or they can be cytokines. Okay, cytokines that are called interleukins. So these cytokines and hormones will tell this pluripotent stem cell in the bone marrow to differentiate into a cell that is required. So if you want to differentiate into red blood cells, there is a hormone that is called erythropoietin. So erythropoietin. This erythropoietin will tell this pluripotent stem cell in the bone marrow to differentiate into red blood cells. So this is a growth and differentiation inducer. If you want to differentiate into megakaryocytes, so these megakaryocytes are what will form platelets. The inducer is going to be thrombopoietin. So thrombopoietin, and it also works hand in hand with interleukin 11. So thrombopoietin and interleukin 11 would tell the pluripotent stem cell to differentiate into megakaryocytes, and megakaryocytes will start budding off into platelets. If you want to produce lymphocytes, okay, so lymphocytes, interleukin 7 will tell the megakaryocyte, the pluripotent stem cell, to differentiate into the lymphoid cell. From this lymphoid cell, then the B lymphocytes and the T lymphocytes are going to be produced. All right. If you want to differentiate into monocytes and neutrophils, okay, you want to differentiate into what we call monocytes and granulocytes. Granulocytes are a type of blood cells. We use an inducer that is called colon stimulating factor GM. All right, so all of these are growth and differential inducers. So for a certain blood cell to be formed in the body, the stem cell in the body has to be told by the inducer to produce that stem cell. That is how hematopoiesis in the body happens. All right. That is how hematopoiesis in the body happens. Now, let's look at erythropoiesis, which is the production of red blood cells. Okay, so this is production of red blood cells. So, in order for red blood cells to be produced, require a hormone that is called erythropoietin. This erythropoietin is produced by the kidney. Okay, so kidney cells. So the kidney cells will produce erythropoietin, and erythropoietin is going to act on a certain stage on the red blood cell production. Okay, so this erythropoietin is the production of erythropoietin. So let's do this. Production of erythropoietin is stimulated by 1. Renal hypoxia. What this simply means is that if the blood that is passing through the kidney cells have a low oxygen concentration, that is what we term hypoxia. That is going to stimulate the kidney cells to produce erythropoietin, and that is going to cause the bone marrow to produce more red blood cells. Number 2, it is also stimulated by prostaglandins. So prostaglandins. Number 3, by epinephrine. So epinephrine. So if you are too excited, too emotional, epinephrine will cause the production of erythropoietin, and erythropoietin in turn is going to increase the level of red blood cells. The last but not the least is norepinephrine. So norepinephrine is also going to stimulate the production of erythropoietin in the kidney. So this erythropoietin will now cause the production of red blood cells in the bone marrow. The maturation factors. Okay, so the maturation and growth factors for red blood cells are very important, these two. So there is folic acid and vitamin B12. So vitamin B12 and folic acid are a necessity. They are required for the maturation of red blood cells. How do they work? These act on the maturation of red blood cells by providing an essential DNA building block that is called thymidine phosphate. So these contribute, okay, or form thymidine phosphate. So this thymidine phosphate forms DNA of red blood cells. So now, if you don't have folic acid and if you don't have vitamin B12, you're going to have a condition that is called pernicious anemia. Okay, pernicious anemia. So in pernicious anemia, we are going to, pernicious anemia is characterized, what is found there is characterized by macrocytic, which means big, or megaloblastic red blood cells, which have a very short, lifespan. So a deficiency in vitamin B12 and folic acid will lead to pernicious anemia. In pernicious anemia, you have macrocytic and megaloblastic red blood cells. So whenever a question comes on pernicious anemia, just know that it is caused by folic acid deficiency or vitamin B12 deficiency. Now, one important factor you need to know is that vitamin B12 absorption in the ileum, of the small intestine requires intrinsic factor. Now, this intrinsic factor is produced by the stomach. Okay, so which is produced by the parietal cells of the stomach. So what will happen is that if there is no intrinsic factor, there is no vitamin B12. So a lack of intrinsic factor is going to lead to vitamin B12 deficiency. This deficiency is going to lead to pernicious anemia. So any condition in the body that is going to destroy the stomach will lead to vitamin B12 deficiency, and it will lead to pernicious anemia. Okay, so if you have atrophic gastritis, meaning that your parietal cells in the stomach have reduced, so the amount of intrinsic factor that is produced is reduced. So that is going to cause vitamin B12 deficiency, which is going to lead to pernicious anemia. So that's one contributor to vitamin B12 deficiency. Another contributor to vitamin B12 deficiency is just lack of vitamin B12 deficiency from the diet. So you're not eating a lot of vitamin B12 from the diet, you're going to have pernicious anemia. If there's a lack of folic acid from your diet, it is going to cause anemia. Folic acid, additionally, is not just important for formation of blood cells, it is also required for the closure of neurotubes in intrauterine life. So if you lack folic acid, you're also going to have neurotube defects. So folic acid is very, very, very important in the body. Okay, that's very important in the body. Okay, very important in the body. All right, so this is where we end for this part of physiology. Watch out for our next video where we're going to talk about red blood cell indices. We're going to talk about hemoglobin. We're going to talk about iron metabolism as well. All right, see you in the next video.